competitor. And basically, our goal is to take the scientific breakthroughs that will be developed and transforming them into social impact by making them more accessible, by building the skills, by providing funding. And also, very importantly, in order to be able to understand if we're being successful or not, we have to measure our impact, and that's part of what I do there. I guess my profile is a mix of economists, data scientists, science policy, and a bit of maybe trying to be a bit of a consultant to be able to understand what's going on across the organization, because I end up getting involved in many very different types of projects. And one of them was this report that we published last year called A New Golden Age of Scientific Discovery. And I guess the reason for that report was that we were as excited as everyone else about generative AI and the potential of LLMs, but we felt among the excitement with that, the potential of that type of AI technology, we're perhaps forgetting another type of AI application or technology that was already creating a lot of impact, and this is AI in science. And basically what we decided to do was to take a bit of a step back from just publishing research, deploying these systems, and actually think about what are the opportunities, where are the challenges, what does this mean for policy, and actually do that by interviewing lots of people inside the organization, like the scientists and the developers who have been creating these breakthroughs. And this is what we published, and it's quite, I guess it was a lucky coincidence that we actually, this was also the year when AlphaFold won the Nobel Prize for chemistry. It's something that happened afterwards, but I guess in some ways demonstrating at least the scientific potential of these models. So I guess like what I'm going to do in this presentation is three things. First of all, I'm going to be talking about the potential of AI for science, and I'm going to be focusing a lot on AlphaFold because it is the system I'm most familiar with. And it's also the system that has been out there more. So actually, we are starting to learn a bit more about its impact. Then, having talked about the high level, sort of like AlphaFold and where it's coming from and how scientists are using it, I'll move much more towards talking about impact, which is where it's more about work I've done or work I've been connected with, which is about how we measure the impact of this technology and what does it tell us about wider opportunities of AI for science, or even wider economic opportunities for AI. And then at the end, I'm talking about some next steps, some policy implications, and very strongly focusing on evidence gaps, which is where we don't know enough about adoption of AI in science and impact, where I would love to get your feedback, get your ideas, and maybe even identify opportunities for collaboration. Okay, so, and yeah, apologies in advance. This is the first time I give this presentation. It is a much longer presentation than I'm used to giving, so I haven't rehearsed it. I don't know how long is it going to take. Hopefully it will fit within the hour, but we'll see how it goes. Let's have an hour. Okay, so the AI for science opportunity. So this is a protein structure. How many people in this room know what a protein is? Okay, so I'm just going to give a very high-level download of what proteins and what we get about proteins, and then start talking about protein structure prediction and AlphaFold and things like that. So basically, proteins are these incredible machines that exist in our bodies. They are the structure of our bodies. They are the workers. They are the wizards that move stuff around. They generate catalyzed reactions. So they are super important. And very importantly, the structure, the shape of a protein determines its function. You can see here hemoglobin, the protein that moves oxygen around the blood. And you can see here like how this is a normal hemoglobin protein, and this is a protein linked to sickle cell anemia. And basically, you can see how the shape of this protein is different from this one because of a mutation. And what this means is that this protein doesn't work. It clumps, it creates pain, it creates infections, it creates all sorts of problems. And the only way we are able to understand the mechanics of this disease is by being able to look at the structure of this protein. So being able to see proteins, like being able to see the world around us and being able to see machines, for example, to understand where they work or not is super important. But how do we determine the structure of a protein? It's not like we can find them around us. They are too small for normal microscopes. And basically what scientists have done traditionally is use experimental techniques like X-ray crystallography to figure out what proteins look like. And basically what this consists of is like shining an X-ray into a protein, crystallizing a protein, then shining an X-ray from different angles. And then eventually you interpret that data, you get a 3D structure, which back in the olden days used to be made of metal, like by ironsmiths working in labs. And then eventually you are able to see the protein. And this one is myoglobin solved by John Kendrew, the chap there, after, I don't know, like 10 years, like working on this. One of the things that scientists figured out by being able to look at the proteins and see their structure is that actually the amino acid sequence, these are like the components that a protein is made of, determine its structure. So that if you have two strings of these amino acids that are very similar to each other, you get a protein structure that is very similar. And basically, what this suggested is that why bother with experiments? If we are able to figure out the function that maps amino acid sequences to protein structures, then we can just predict those structures with amino acid sequence, and we don't have to spend 10 years running these experiments and building all of these complicated metal structures. And this is where the dream of protein folding comes from, taking amino acid sequences and using them to predict protein structures. There's like a big need for this, like to be able to like create these protein structures, because we have lots of amino acid sequence whose structure we would like to be able to see. What you can see in this chart is simply showing the evolution in the number of sequences, protein sequences, that are, have been sequenced, have been collected. And basically around 2024, it was 250 million protein sequences that we would like to visualize, what we would like to see the structures. One way to do this would be to develop some sort of theory of how an amino acid sequence become a protein structure. But actually, this is super complicated. Basically, what you can see here is how this works. Basically, an amino acid sequence will explore this energy landscape and eventually find a stable configuration where it acquires its shape. So this is something that is quite difficult to theorize about in a principled way. It's almost like mathematics are not enough to be able to model this. It's just too complex. And this is what, for example, our CEO, Dennis Sasavis, says that it's like a problem so complex that we need to, for example, use tools like AI that are able to capture that complexity much more effectively. And this is, I guess, what we have been exploring. And yeah, this is a broad-based challenge. This is not a challenge that is specific to structural biology. We have the same problem in genomics. We have the same problem in physics. We have the same problem in chemistry. We have the same problem in weather. In all of these areas, we have a lot of data. We want to model very complex phenomena. And we need to develop analytics technologies that help us to do that. And this is what AI brings. No, I guess it's not connected to my laptop. So far, I'm talking about why scientists would want access to AI to be able to deal with all of these like large-scale data sets and all of these complex phenomena. But the question is also like, why would economists, why would other people be interested in the use of AI in science? And I guess like the first observation here is that better predictions, for example, about protein structures are useful. And this is actually a chart I stole from Ryan's wonderful research, which basically what it shows is that the better pictures of proteins you have, the more drugs you tend to develop around those protein structures. And this is because you can figure better what's going on with that protein, and you're able to develop better drugs related to it. So, and we also have, for example, research showing that the Protein Data Bank, which is this database where scientists put their protein structures, around like 90% of the drugs approved by the FDA between 2020 and 2016 were informed by shapes, by protein structures from that database. So it's like a very big deal for drug development to have these protein structures. The problem is that producing them is costly. And again, this is something I stole from Ryan's paper. Basically, what this is showing is how long does it take for scientists to, once they have collected experimental data about a protein structure, to then create the protein structure. And basically what this says is it's on average is 2 years. 2 years from collecting experimental data until you have analyzed that experimental data and created a protein structure. And actually we hear anecdotally lots of people saying that their PhD would be solving a single protein structure 4 years or so. Just think about if you have to solve those 250 million sequences that I showed you before, how many years, how many billions of dollars it costs to basically be able to create those structures. And what this means is that in 2020, when we launched AlphaFold, there were only around 170,000 protein structures solved. This is less than 1% of like all of the amino acid sequences that we would like to be visualizing that I showed before. Again, I'm telling you about AlphaFold, but this is not specific to AlphaFold, right? This is like in general, like a sort of science. We have situations where we would like to have better predictions, better data. Generating these predictions, generating this data is very expensive. And actually, if we found ways to accelerate this, to improve the productivity of this process, this would be beneficial, would generate lots of economic benefits, and might help to deal with the slowing down of science that so many of you in the audience have contributed to evidence. Ryan?
One thing that I guess Carol and I have been thinking about, maybe you have thoughts on as well, is that, you know, scientists have been doing crystallography now for 40, 50 years or more. And, you know, the sort of growth of the PDB, especially through the 80s, 90s, 2000s, was pretty large. And I think you could imagine that also was very selective. Like scientists were choosing the projects to solve experimentally that were maybe the highest potential, right? Those proteins like the hemoglobin that were relevant for disease study or therapies or other, you know, scientific import. And so one sort of concern might be like there are maybe, you know, 90% of, or more of all proteins that are unknown, the structure, and we can predict it. But I guess how do we think about whether those are very useful for us? And have you put any thought into trying to think about how to sort of measure that or quantify the sort of relative importance of the unsolved structures compared to the ones we've already done?
Yeah, there's like maybe like the first observation there is that there is a bunch of proteins that, or amino acid sequences that we were just not able to solve using the existing methods. So there's like a bunch of like almost like gaps in this map of the protein space. So it's almost like developing tools that accelerate the productivity of scientists trying to do this, create almost like proteins that were not available using different, like couldn't be solved using available methods. The second observation is that it's almost like it's an empirical question whether database tool like AlphaFold actually has moved the dial, has like changed the behavior of scientists in a way that suggests that the predicted protein structures were valuable. And actually, I'm going to be providing a bit of evidence about that later. All right. So this is like the beautiful architecture of the AlphaFold 2 system that our science team developed in order to tackle that scientific problem, that economic problem of solving, being able to predict protein structures. Basically, what it does is it takes an amino acid sequence. It does a bunch of quite complex things with it, and it generates a predicted protein structure. And something that's super important about it is that it doesn't just split the protein structure. It tells us when it is highly confident about the prediction and when it isn't confident, which is going to be very useful for scientists. A couple of things to say about it is that it is trained on the Protein Data Bank, the database I mentioned before. it is using various structural biology data sets as it goes. And also this is quite cool, actually. It's using its own predictions, the high quality predictions it generates. They are feeding back into the model and they make it better. So it's like a really cool, I think, the combination of like biology domain knowledge of being able like to what data sources to work with from that domain with it's actually the transformer architecture, like from like AI, and there's like all of these cool tips and tricks to make it work. And I guess this is the model that was underpinning AlphaFold 2. We published a paper in 2021. We created a database to release the structures. And what the model did was massively improve on the performance of protein structure prediction systems compared to what came before. And this is based on the results from the CAS competition. I'll be talking a bit more about that later. And then it also resulted in the Nobel Prize in Chemistry for Dennis Sassabis and John Jante, who led the development of the model on our side. So I guess so far, I guess like this has illustrated a bit like what AlphaFold is and why it's important. What we try to do in the golden age of discovery, the SA I worked with, was try to almost like take a step back and think not just about AlphaFold, think about other AI models in science, and think about what are the opportunities that they bring. I guess like thinking about it as high level, I feel that almost like what they do is bring ICT economics to science. We're investing on building a model, collecting data to train the model, and once the model is trained, it's actually be able to generate predictions at a very low marginal cost. So not like digitalizing experiments, and in some ways this is what AlphaFold does. It's basically able to create 250 million protein structure predictions with high accuracy, and then basically these experiments are themselves a source of data that can be used in further analysis. And actually this data is a source of signal that might help us to develop better solutions. For example, when thinking about developing a drug, what are the drug molecules which are better suited for a particular protein we want to target. So it actually helps prioritize and develop better solutions. And there's like other ways in which AI can help, which are maybe a bit less related to AlphaFold. Like for example, transform how scientists digest and communicate knowledge. And also like, for example, be able to develop, I guess, complex, like models of complex systems and the interaction between the components. I'm not going to be talking about that so much, but we really dig into it in the report if you're interested. So I guess this leads us to think about, okay, so where do these AI science breakthroughs come from? And basically in the context of the paper, what we do is basically talk about this idea of the AI for science production function, which is basically a collection of inputs and activities that help us to develop these AI for science breakthroughs. I'm not going to be talking about the whole thing. We get into a lot of detail in the report, but I just wanted to highlight the starting point of this production function, which is about the problems that we're trying to solve. And basically the question that we always ask ourselves at Google DeepMind is whether a scientific problem is ML-shaped. And basically what this means is this a problem where there's a lot of data, or where we could generate data, for example, synthetic data that we can use to train a model? Are we able to evaluate the model to figure out if it's improving or not? To do like this thing we call hill climbing, which is find a metric that we want to improve and basically like innovate in order to go up that hill and get signal about whether we're being successful or not. But also very important for us is this idea that you could use these methods to look at many different types of problems, but we prefer to focus on these things we refer to as root node problems, which are almost like scientific problems, where if you solve that problem, you unlock lots of opportunities for download research and creating impact. And I guess these are the problems that we try to prioritize, and AlphaFold is one of them, but we think that there are many others. So I just wanted to just mention, before I finish this section, a couple of inputs that were very important for the development of AlphaFold. And I guess they illustrate the importance of public goods for AI and science. So I told before about data. AlphaFold was trained on the Protein Data Bank. This is a repository of protein structures created in 1971, where scientists who basically use experimental methods to create a protein structure, they will deposit their structures there. And they do it in a standardized format, and this is validated. And basically what you see here is just the increase in the number of protein structures which are deposited there. I think I said before, 170,000 or so around 2020. And what's very important about the Protein Data Bank is that it's not just a place where scientists put protein structures. It's also a collection of institutions and social norms about what it means to be a structural biologist. In order to get funding, in order to get published, you need to contribute your protein structure to this repository, which creates very strong incentives for structural biologists to do this and helps us to create this very valuable data set. The other important input, I mentioned before evaluations and benchmarks. I feel like one big problem with AI models is that it's very easy to cheat with AI, which is what happens when you put the thing you want to predict on your training data. And then the model is able to trivially predict that, make that prediction. So you need to have very rigorous methods to be able to evaluate whether these methods are performing. And this is what CAS gave us, is the critical assessment of, I've forgotten what the acronym means, critical assessment of protein structure prediction. And basically what this is that once every 2 years, it's almost like Olympics for protein structure prediction leagues. Basically, there will be a number of amino acid sequences that have been solved. We know what the structure is through experimental methods, but the structures haven't been published. So actually, the amino acid sequence is shared with the competing teams, and they will feed them into the models to make predictions about the structures. But it's not possible for them to cheat because the structures couldn't have been part of the training set because they haven't been predicted yet. And then basically, the organizers compare the predictions with the actual real protein structures. And basically, what this makes it possible is to have a very rigorous method for the assessment of these models. And this is what, and basically in CAST 14, what this is showing is a distribution of performance from different entrants, and this one is AlphaFold. The CAST almost gamified the process of protein structure prediction, and also gave us a very rigorous signal that AlphaFold was really performing well because it was learning something about the structure of the connection between amino acid sequences and protein structures, and it wasn't cheating. And actually creating these benchmarks and maintaining this kind of institution for rigorous assessment of AI models for science, I also see this as a public good. It's also about public goods in outputs. And here, I guess, you could develop the best model, AI for science model. You create this breakthrough, is something that gets you a Nobel Prize. That's knowledge, that's a public good. But there's a question about how do you make that knowledge even more accessible? How do you release it so that more people can use it? And this is something that's been very important for DeepMind, and this is actually the kind of work my team does, which is in addition to releasing the model and releasing the paper and releasing the code, we also created a database where users can explore 200 million protein structure predictions. And it's almost like a Google search for protein structures, so that structural biologists can come here, they can search for a protein, they can look at the structure, download it, and use it in their research without having to be coders or without having to install, say, the AlphaFold code base in their servers. I think this is really important, is this idea that we need to actively try to increase the accessibility for our models in order to make them impactful. This is something we have spent a lot of time trying to do. Okay. So this is the end of the first part, which was very much about what the wonderful AlphaFold team accomplished and why we think it's important. Now, I'm going to be talking a bit more about the evidence of impact, which also will connect with the question that Brian asked about, so what? And basically, the thing here is that AlphaFold 2 is a massive shock in the supply of useful scientific data. This circle here is all of the proteins that we shared through the AlphaFold database I mentioned before. And these are the ones that we had before. So it's like this big, massive increase in the supply of data. The question now is, what are scientists doing with it? And is it useful? Is it valuable? And this is what I guess people like myself and my team coming to try to measure this. Yeah, my team, just very quickly, I guess our mission is to accelerate and communicate the impact of our technologies using data and evidence. We are very focused on basically, say, if you think about the ways in which people evaluate AI models, some of them evaluate capabilities, which is like you basically see if a model performs well against a benchmark. Some people are evaluating them by running experiments, like to see what's the impact of those models when they are deployed in a pilot or experimental setting. And then there's people who look at the systemic impact of AI by looking at post-deployment impacts. And this is the kind of work that our team does. And I guess this is very much informed by Denis's view that we should use the scientific method as we are approaching this very pivotal moment in human history. In other words, we need to get scientific about how to track the impact of AI, including AI in science. I mean, this is just like some KPIs, around like the number of people who have used the database, the citations in the paper, the number of countries using the database. And I guess the idea is that we are using psychometric data, we're using product analytics, for example, from the database, using LLMs and collaborating with external researchers to try to generate this evidence. And I guess here is where I emphasize this idea of collaborating with external researchers, because it's one of the reasons why I'm so excited to be talking to you today. So I hope that we can collaborate in some of these analysis. Just before getting into some of the findings, there was like a bunch of measurement issues here that we need to be very careful with. The first one is that we want to identify AI developers and users. In the case of AlphaFold, actually, lots of the research that is being impacted by AI, by AlphaFold, is not scientists developing AI or talking about AI. It's just people who are using the structures that we predicted. So we need to be able to track that. And also the question of how do we track downstream influence and impact. We know that the people who are going to be using these protein structures at the beginning are going to be more like biologists. And then if we're interested in the real world impact that this has, for example, on biomedical research, technology development, we need to offer the downstream. And there's a question about how we track those downstream impacts. I guess one way in which we have tried to do this is by analyzing, say, looking, for example, at citations. It's received around 35,000 citations, and actually analyzing the full text of papers in order to understand whether those papers are actually using AlphaFold, like in what way are they using it, and then also trying to create citation chains that don't just consider those papers that are directly citing AlphaFold, but those that cite papers, and then think about how do we use information about the citation intent of papers connected with each other in those chains in order to avoid that situation where we are trivially expanding the impact of AlphaFold just by looking at everyone who cited a paper that cited AlphaFold. So that's something that we're trying to do. And I guess what this shows, for example, this is a chart from some work in progress from some researchers we have been working with. It's basically that most of the research that is on AlphaFold is downstream. Is not the papers that cite AlphaFold directly, it's the papers that cite AlphaFold. Actually, what's interesting is that if you look at the papers that are just citing AlphaFold directly, they are in biology. The ones that are citing it further downstream are in medical sciences, are more applied papers, suggesting that it's almost like this linear model of scientific research, informing applications, and hopefully eventually informing impact. The second thing that is very important for us is that we don't just want to track how many papers are building on AlphaFold or citing it. We actually want to identify individual use cases, right? Some like types of applications. We are, for example, interested on applications of AlphaFold related to antimicrobial resistance. or applications of AlphaFold related to cancer. And basically, obviously you could do that by using mesh, looking at mesh terms and things like that, but something else that we have tried to do is to leverage large language models to start to build taxonomies of use cases of research using AlphaFold. And I don't know if you have read, if you're aware of like some research by Anthropic called Cloud Insights and Observations. We have done something similar where we use LLMs to basically build hierarchy, like cluster hierarchies that make it possible to explore the research that cites AlphaFold at different levels of resolution. And then what's really nice about this is that in addition to giving us like this thing, you can't see anything, but it's basically like a distribution of papers over different use cases. This is useful like for us for analysis. It's also something that we can use to identify scientists to engage with in order to learn how they're using AlphaFold. Obviously for our Fonts team, they're interested in developing impact studies and things like that. And this is where we are able to use some of like this citation data for those use cases, as well as the more analytical ones.
Yeah. So just a clarification on this, the ones that you call AlphaFold related papers, how do you tag those? Is that based on they're citing the database specifically, or they're citing the jumper et al. Paper, or how do you?
So there's like an AlphaFold corpus, which includes the jumper paper, the AlphaFold multimer paper, the AlphaFold database paper, and now the AlphaFold free paper. And we're basically interested in any papers that cite any papers in that corpus. And then, what we do is analyze those papers to figure out the citation intent, which of them are actually using AlphaFold. And then we look at the papers that are citing those papers, where again, the citation intent is around using the methodology or using the findings rather than citations in the introduction or in the conclusions, which is more likely to be a tokenistic kind of citation. And that allows us to create citation chains that hopefully are capturing work which is more likely to be building on AlphaFold than just like cheap citations.
I mean, many of those papers are like computer science applications of similar neural net algorithms in other applications or something. So they're not sort of like directly related to protein science, but they're saying, here's a tool that we like and that we can sort of apply to our own application. Do you think of those as being impact of AlphaFold, or are you more narrowly focused on protein science applications?
So I feel like if someone developed like some amazing kind of like new AI model, completely unrelated to biology, that was significantly building on the AlphaFold architecture, that's something that we would be interested in tracking, and actually that illustrates the general purpose nature of this innovation. As it is, the reality is that most of those, like say archive papers citing AlphaFold, they are almost like exclusively tokenistic, kind of like citing it as an example of an interesting AI application, but not really building on it in any kind of substantial way. Okay, so again, like maybe this connects like in an anecdotal way to Ryan's question about what's like the added value of like say the protein structures like predicted by AlphaFold. This is just like 3 examples of how scientists are using it, and this is hopefully giving you a bit of a context for some of the findings which are going to be coming later. The first thing that scientists are doing with AlphaFold is actually to accelerate experimentation. So actually, AlphaFold hasn't arrived to make experiments unnecessary, because we still want scientists to run experiments to figure out the structures of proteins, because first of all, those are useful for validating the protein structure predictions, and then they can be better quality, and they actually generate more data that we can use to train better models. And actually what AlphaFold has done is really help scientists interpret experimental data and run those experiments faster. And a really nice example of that is where scientists use AlphaFold to solve the structure of this thing, which is the nuclear pore complex, which is like this very complex, like protein complex that manages like the flow of different materials between the nucleus of the cell and the outside. They're also using AlphaFold to develop biological hypotheses, which is where they say, oh, this protein is this shape. This means that it might interact this way with this other protein, and this is telling me something about how this is work, or how I might be able to design like a new molecule that might be useful in some way. And a nice example of this is where scientists use AlphaFold to design almost like a nano syringe that is able to inject proteins inside cells that might be used like for example, to delivering therapies inside cells. It's somewhat like the microanalysis, and then you also have like the macroanalysis, which is where scientists will say, oh, we have 200 million predicted protein structures. Can we analyze them at scale in order to figure out something about the protein universe? And basically, what these ones did was cluster those 200 million proteins into different clusters and trying to figure out which of them have been annotated before, were there any unexpected relations between different clusters, and it's almost to learn about what is like a snapshot of the protein universe told us about evolution and the relation between different organisms. And you see it's like different types of analysis enabled by this tool.
Juan, can I ask a question?
Of course.
So AlphaFold enables new kind of questions, new hypotheses that scientists can be asking because they have new data, right? There's also an AI co-scientist paper that Google put out a couple of months ago, and I imagine, and that is not about the humans asking, coming up with more hypotheses, but it's that the computer is coming up with more hypotheses. And so I'm curious to what degree that's also enabling like an AI scientist to come up with more hypotheses versus a human. Is there are you seeing some kind of differential between human hypotheses versus computer hypotheses based on something like AlphaFold data?
That's a really interesting question, and with so the AI co-scientist, and I'm going to be saying something about it in a bit. It is currently only available for trusted testers, so it's very early days for it. I haven't been involved with that project yet, and so I don't know how people are using it. And it would be super interesting to be able to compare those hypotheses coming from co-scientists with those coming from human scientists. I guess maybe one thing to say, at least about what was published in the co-scientist paper, is that co-scientists work within quite narrow kind of like domains that were set by the scientists. They told it, I want you to develop, identify what the story purpose for this problem and maybe focus on this area, and then it went over and did it. So I think it was still quite narrow creativity of problem solving within an area defined by the scientists. Something that's quite interesting about co-scientists is that it's able to use AlphaFold as a tool. So actually, it's going to be quite interesting not just to see what questions it comes up with, but also how it's using these kinds of tools and models. Okay, so what I'm going to be doing now, and so Ryan, you said until quarter 2-3.
Yeah, we got a half hour left. All right.
Yeah. So I'm just going to give you emerging evidence of, I don't know if I want to call it impact, because I think that's causality and even higher than that very causal now. Emerging signals of the changes that we're seeing in structural biology that correlate with AlphaFold adoption. And I'm going to be talking about changes related to productivity. I'm going to be talking about changes related to innovation. I'm going to be talking about the distributional aspects of those changes, and then say something about externalities. So we're going to be covering a bunch of different things. And I guess something that's very important that we do a lot of in the golden age of discovery paper is I'm going to try to say something not just about the benefits, but also what we're learning about some of the risks and some of the challenges, and whether those are warranted or not, based on the evidence we're getting from AlphaFold. Okay, so productivity. What is AlphaFold's impact on the quantity and quality of research? So basically, we have this paper, this working paper by Ju that came out last year. And basically, what he did was to compare structural biology to non-treated fields in the life sciences. Interestingly, it found limited evidence of impact on the amount of papers that structural biologists publish compared to the non-treated fields. But what it found was an increase in the citations received by structural biology since the arrival of AlphaFold compared to the non-treated fields. I think it's quite interesting. This is comparing the changes in citations. And actually what he finds, which is going to be linked to the discussion about distributional aspects later, is that in general, it is like scientists and labs that have more experience and more skills are the ones that seem to be benefiting more from AlphaFold, and those labs that don't have that experience, actually, they barely see it. It suggests that there's like some complementarities there between the AI predictions and the skills that exist in those labs. This is the research that I mentioned before, which is relying on this citation change. And basically what they do here is they compare AlphaFold, like basically labs that are citing using AlphaFold with a structural biology baseline, and they do matching on like a bunch of like variables in order to try to ensure comparability between like the labs they are looking at. And they also compare the changes experienced by the labs that adopt AlphaFold with the changes experienced by labs that adopt other frontier biology technologies, both involving AI and without involving AI. What they find is that the research labs that are adopting AlphaFold tend to submit more experimental structures. So this is the idea that AlphaFold is accelerating experimental structural biology. And also, I guess like there's maybe like a quality filter there in the sense that these are protein structures which are submitted to the protein data bank, so already are like checked in some way for like the quality. And similar to the finding I mentioned before, the labs adopting AlphaFold also tend to receive more citations. And actually, this is quite similar to the uplift in citations experienced by labs that adopt other counterfactual technologies. Yep.
I mean, so experimental structures going up in quantity, but output in the first study, I guess, is publications not going up. How do you reconcile those two things?
Because actually, there's going to be like lots of biologists who are using AlphaFold for other types of biological research that doesn't involve submissions of structures to the PDB. So basically, this is almost like a heterogeneous effect within a subgroup of structural biologists.
I see.
Yeah.
So the overall quantity has been going up because it's a lot of things that aren't protein structure.
Yeah.
Okay.
And this would be actually, these groups, they are very important. They would be a small fraction of like the overall structural biology community, because they are the ones who are working with experimental data and do all of the things I was talking about before. And finally, actually, there's some evidence of downstream impacts in terms of like citations to research that builds on AlphaFold from clinical articles as well as patents. And yeah, like the final bit of evidence I mentioned here is more qualitative, and this is coming from like a paper we published in PNAS last year, which was a bit more of a literature review of how structural biologists are using AlphaFold. And I guess something that we found that maybe speaks to the idea that AlphaFold hasn't brought with it like a massive increase in the supply of low quality research is that there seems to be like quite rigorous, strenuous validation of AlphaFold predictions by the community. And also, it's been super important that we are sharing the confidence, like the confidence about predictions together with the structures. And again, this is helping scientists to interpret the structures and whether they can use them to build on the work. Okay, creativity. I guess like the question here is, it's a very important one. Is AlphaFold encouraging incrementalism or is it encouraging innovation? You know, if AlphaFold is just encouraging scientists to focus on those areas where there are better predictions, maybe it's actually leading to a narrowing of research. And I guess what we find here is like the comparison of protein structure submissions, where it compares, like say, compares the labs that are using AlphaFold, even with other counterfactual technologies. They tend to involve like the submissions from labs using AlphaFold tend to involve more novel proteins that weren't like hadn't been solved before. And also we find some evidence that those protein structures seem to be more different, like from those that were had already been submitted. So like using various like measures of novelty in structural biology, like TM scores and things like that. Which is suggesting that AlphaFold is encouraging scientists to look at new questions and new areas. And there's also research by Gene forthcoming, which who are also looking at this. And this research suggests that the papers citing AlphaFold are more likely to be studying novel genes, functions, and organisms. So it's all of this is suggesting that AlphaFold is potentially, very like my interpretation of this, is that in some ways by, and this is informed by a recent Josh Camp paper on the impact of AI in scientific discovery, that in some ways AlphaFold is commoditizing the more incremental types of research and encouraging scientists to maybe like cast their net farther or try like to push the frontier. Distributional aspects. So I guess for us it's super important that the benefits of like a technology like AlphaFold are widely shared, and we would like to see scientists from all different countries like using the technology and benefiting from it. So this is something that we have explored. And I guess here, on the one hand, we feel, okay, we're making all of this freely available. It's free. You just go to the protein, like the AlphaFold database, you can download everything. In principle, this could be democratizing structural biology research. But also we know that the reality is that scientists in different countries, scientists in different communities have different skill sets, access to different resources, or even information about AI that might prevent them from using AlphaFold. And actually what we find in our own internal analysis is that scientists from low and middle income countries are less likely to use AlphaFold than scientists from rich countries. And this is just like a logistic regression where the dependent variable is whether a paper is AlphaFold or not. And we include a bunch of predictors, including whether the paper equals LMICs, and we have a negative coefficient. However, we also find that AlphaFold is more likely to be used to study neglected diseases and malaria, and basically diseases that create big impacts on low and middle income countries. So even if the scientists are not using AlphaFold in LMICs, actually scientists in other countries are using AlphaFold, are more likely to be using AlphaFold to study those diseases, which maybe connects with the previous observation about how AlphaFold is encouraging novel types of research. Finally, expertise. So it's not just about distributional aspects of AlphaFold or impacts in relation to like the geography of scientists, but also whether they are knowledgeable or not. And as I already mentioned before, highly cited researchers are more likely to adopt and benefit from AlphaFold. And here I'm I guess like there's many different mechanisms that would explain this. One of them is that they simply have more resources, they have more skills, they have more ability to adopt this new technology. Another one is that AlphaFold might be encouraging scientists to explore new areas where the kind of skills that these labs have are what's required to do with research. Or it might be that these scientists have more incentives to I guess to adopt AlphaFold, especially as the more incremental parts of structural biology get commoditized by the structures. And I would love to us to have a better understanding of what mechanism is driving this.
In that Aidan Toner Rogers paper, which is about GNOME, or maybe it's not about GNOME, but it's about some machine learning material science predictor, has a similar finding where the kind of most productive, and that's a randomized controlled field experiment source, but it's very similar in terms of the highly productive people benefiting the most.
Yeah, and it's also like the research from Matteo Tranchero, who's looking at GWAS, not AI, but it's like a similar type of technology. Again, very strong evidence of complementarities between domain knowledge and the ability to interpret like AI predictions. I guess suggesting that people who don't have that domain knowledge end up being like bamboozled by false positives. And yeah, like the Julian Richardson, the other like forthcoming paper finds something similar. Finally, externalities. What are the indirect impacts? And I guess this is where you create knowledge, to create a new technique, you make it open source, what happens. And basically, the first of all is the idea of helping to develop new methods. This is like, so the competition where AlphaFold won was Cas14. This is Cas15, 2 years later. Google DeepMind didn't enter, but the top 3 participants, all of them were building on AlphaFold because they could just take the code, build on it, improve it, like innovate. So in some ways, like the technique becomes part of the, I guess, like the furniture of structural biology and preventing structural prediction. And that's something that's very important as well. Like I said before, AlphaFold is helping to develop new run experiments, creating more data, and this data then becomes training data for like new models. So again, like, you create like a positive feedback loop there. But maybe something that's really difficult to it's a very kind of passive benefit, but it's this idea of inspiring AI in life sciences as well as other parts of science. And maybe like in GDM, anecdotally, I feel that the fact that AlphaFold has demonstrated that you can get a Nobel Prize in the scientific discipline using AI is making everyone think like, oh, what are we going to get the next one? You know, like getting a sense of like scientific excitement about using AI in science that maybe wasn't there before. And yeah, like around the environment, like there's like this obviously this very big concern that you are training these models, they're consuming a lot of energy, they're consuming, this is generating emissions. So I guess what are like, that could be a negative externality from these models. But I guess at least in the case of AlphaFold, the baseline is using synchrotrons, which is like very like big like energy beams to create X-rays that were used to generate experimental data. And actually, when we compare the energy consumed from training AlphaFold with the energy consumed creating the experimental data, we find that training all of AlphaFold consumed as much energy as creating experimental data for 10 protein structures. So it's consuming energy, but it's relatively efficient compared to like the traditional experimental methods. And that's something that we always need to remember when we talk about the environmental energy costs and environmental impacts of AI. It's like, what is the baseline? How much energy would we consume using an alternative method to achieve the same scientific goal? So my recap. So I guess like the first thing to say here is this is very early evidence. This is mostly descriptive analysis, some correlations. This is something coming from someone who works in GDM. Like, obviously, I might be biased. I'm not. We try to be very, very rigorous and independent, but this is just one view. And I guess one of the things that we really want to encourage and would like to see much more of is like more independent contributions, more people looking at these questions from the outside and improving the evidence base, making it more rigorous and figuring out some of the areas of uncertainty or like areas where we still have open questions about mechanisms and things like that. But bearing all of that in mind, still I feel like this is a positive impact of a positive impact story. It's been enabled by the fact that in structural biology, there's many different methods that people can use to validate AI predictions. They can use experiments. They can also like develop a hypothesis and test it downstream in the wet lab. So there's many different ways to triangulate. The fact that AlphaFold, I guess like using like the comparison between floodlights and street lights from Abhishek Nagaraj was a floodlight. It literally created like a map of the whole protein universe and maybe created less risk of skewing researchers towards focusing on specific like narrow areas. And also like around understanding, I think like super important for me is that even though AlphaFold in some ways is a black box about the mechanisms that are driving or like the process through which a protein, like amino acid sequence falls into a protein, is still creating data that is enabling like downstream improvements in understanding, for example, in the biology of disease and things like that. And yeah, just to say that if we are going to be encouraging widespread adoption and making the benefits are widely shared, this is going to require a lot of investment, and GDM is providing training, funding, and compute to reduce adoption gaps. So we're also trying to find ways to encourage, for example, scientists in LMICs to adopt AlphaFold and create impact from it. But I guess like at the end of the day, what's important for us is like this kind of like testimonials, impact stories, like examples of how, for example, in this context, like AlphaFold is helping to accelerate research that might help to develop new antibiotics. So I think at the end of the day, this is what we really are really excited about, is enabling those use cases and that kind of impact. Yeah, so I guess the whole presentation has been super focused on AlphaFold. Like, and if you read the paper, like the golden age of discovery, actually AlphaFold is just a very small part of it. But we feel that AlphaFold is not a one-off. I would feel that there's like amazing opportunities to use AI in many other parts of science. Many of them are already being explored by GDM, some others are not, like genomics, like weather, like materials, like matter before, like agents, like I'm developing like actual systems that are able to develop their own hypotheses and pursue them with more autonomy. So like so many opportunities to use AI in science that maybe don't give you a Nobel Prize, but probably give you a lot of like impact and lots of beneficial applications. And we're very keen for those to be explored. So, yeah.
Just curious about the weather one, actually, just because I think of the other applications as sort of understand how you would take known structures that are true and repeatable and then make predictions in an algorithm. Whereas weather, I think it was a classic system, chaotic system.
Yeah.
Just like a fundamental, the math is working against you in a very hard way. So it was not so obvious to me that that would be a great target for AI. So I mean, is it a very different model or is it sort of the same model? It's just, how do we think about that?
So actually I haven't read the GenCast paper, which is the last one, but I know that the GraphCast paper is like a graph neural network. So it's like a network that encodes like more like facial like relationships. I think that's the way they have managed to make that work. But you know, like with every other example here, it's been reliant on amazing data sets created by the European Center for Mid-Range Weather Forecasts, as well as like the development of like really interesting benchmarks that are almost like these like amazing scorecards where we have like many different variables being scored, for example, at different heights, like in the atmosphere. But yeah, they made it work. And the follow-up GenCast is actually about using that to generate ensembles of predictions that are more useful for understanding, for example, the evolution of extreme weather events where there's a lot of uncertainty. But yeah, I think I will be able to get into more detail beyond that. But yeah, just to say, this is an example of like the using AI to model complex systems kind of opportunity that I didn't say so much about before because it was less related to AlphaFold. Okay, so we have 10 minutes just to conclude. So in the essay, we talk about some of the policy implications, which is, okay, how do we make more breakthroughs like AlphaFold? And how do we create more value for those breakthroughs that we already have? And basically, we talk about the importance of problem selection and some of like galvanizing or bringing together the community around like areas that we agree are important opportunities to use AI in science in the same way that Hilbert did it for problems in mathematics at the beginning of the 20th century. Then it's a question about data and how do we basically increase the quality of the quantity of data, the quality of data, and the accessibility of data. There is skills, and this is both about equipping scientists with the skills to become model developers, but also become users of models, because that's going to become increasingly important as part of their work everywhere. And obviously, the one that I'm really it's very close to my heart is on the evidence and experimentation, which is how do we build the evidence base about AI adoption and impact, and then also how do we encourage experiments with new ways of organizing science that I guess like harness the potential of AI. And yeah, I feel like this presentation has illustrated that it takes a village to deliver these kinds of breakthroughs, and it requires a lot of collaboration between industry, between academia, policymakers, and also civil society. So I guess that's one of the reasons why I'm so excited to be talking to you and potentially identifying opportunities for collaboration with your community. So we are working on a follow-up essay to the Golden Age of Discovery essay, which is about AI for science, meta science. In other words, what do we know? Yeah, basically, what is the evidence base for the adoption and impact of AI in science? Basically, what we want to do here, maybe in the spirit of problem selection, is identifying some evidence gaps that we feel are very important, where we would like to see work to fill those evidence gaps. And this is something that we'd like to see the community do, but also like it suggests potential areas for collaboration. And I guess around adoption, I still don't know very well what are the levels of adoption of AI in science. This is going to become harder. People increasingly use models and agents, including to develop new hypotheses, and might not always be citing them. Increasingly, AI agents might themselves start to produce papers. How we're going to be tracking that, as well as the indirect impacts. It's not just about who's developing the models, but who is using the predictions generated by the models. And also, super importantly, what are the drivers and barriers? So something I mentioned before is what are the mechanisms driving increased adoption by labs with like high levels of expertise or capability? Outputs and processes. What is the impact of AI on creativity, reliability, understanding? Do we start losing at some point if we are building science on black box predictions? What does that look like? How do we track it? How do we address it? And also things like job satisfaction, like the Tony Rogers paper around AI for material discovery suggested that AI was making the scientists in this company more productive. It was making them more creative, but actually they didn't like it. It made them feel like that their skills were devalued and make them feel less happy with their job. So I think that contrasts with like our experience, like engaging with scientists. We use AlphaFold. And I guess it would be great to get a sense of like what's like the evidence, including a more systematic evidence based on that situation.
As I read that, I remember it being a bit that the AI was taking over the fun part of the job, which is kind of the blue sky thinking about a new possible material and what its properties might be. And then it was sort of turning people into just sort of experimental verifiers. But I could sort of imagine with AlphaFold that it's taking over this hugely experimental part of the job and allowing you to be a bit more thoughtful about what it does. Kind of the reverse, maybe in a sense, or like you're verifying, but you're also getting to, think a lot more about function more quickly, and are spending less of your time just taking more of your time thinking about which proteins would you really want to investigate and why, et cetera.
That's interesting because in some ways it's like this more equilibrium effects of what do you do with the extra time you get from like a tool like this, and maybe when you're working in a company, what you do is more of the same thing. Whereas maybe if you're like a scientist working in academia or in a lab, you actually have a scope to maybe have a bit more agency or autonomy about what you do next, and maybe you can focus in other areas that just be more creative.
I will say many of those scientists are so highly trained, like much of their thought process was like how to make, how could I possibly crystallize some new protein, or like they would spend much of their time theorizing about the actual methods, and much of that could be displaced fully by this, and they would have to do something they're not trained in.
So in some sense, also creativity is overrated in the sense that scientists are driven by achievements, sort of what is the thing that they achieved. So if you like look at like structural biology, like just the structural proteins, for every issue of nature, there's at least one paper about finding just the structure. I mean if that then is automated, then it almost doesn't matter if you're like more thoughtful about what can we do now with this structure, because finding the structure I can see is like a huge achievement. And then all of a sudden that bar just disappeared, right? In a sense. And so
I mean, how much longer are we going to reward it? The experimentalists are still they're like using AlphaFold in part as sort of a model for how to like successfully solve a structure experimentally. But how much longer are science and nature going to find that to be, worth the page space if they're drawing a lot on what AlphaFold has already predicted?
Right. Yeah. So that's where, for scientists, I'm just thinking, for trying to find a structure is tedious. Actually, some of the labs are very famous from Chinese scientists too. It's extremely tedious work, but then it's very it's high achievements in the sense, in the past. But
I guess like in some way, one of the things that they will do is like focus on those areas of like experimental structural biology where actual predictions don't perform as well. We have seen a lot of like focus on basically this idea of like intrinsically disordered proteins, which are like proteins that change conformation, change shape, depending on say what the molecules they're interacting with. And actually by definition, it's quite difficult to predict that because there's not a single good prediction. Like actually, those predictions with AlphaFold just look like low confidence, the parts of the protein which are wiggling around. And we have seen a lot of activity in that area because it's one that where the AI model doesn't perform so well. So like the biologists, we focus towards that area, maybe where they have a comparative advantage compared to the AI model. I feel like this conversation really brings me to like this one, which is like a massive one, and we're discussing it more before, like what we just don't know enough, which is what are the impacts of AI on scientific labor markets? So actually, in a way, the conversation we are having now is about labor displacement, is as for displaced experimental structural biologists. And this is something that on overall, we are still seeing lots of protein structures being submitted to the PDV. We haven't seen like a complete collapse in the like applications to structural biology degrees. We're still seeing like a lot of funding going in this area, at least like in the last couple of years. But that, for example, scientists moving around, like are they leaving like the discipline? Are fewer scientists? Do we need fewer scientists to create the same like level of output in terms of like the protein structure submissions to the PDV? And I feel like this area where we just don't know, and it would be great to find out more. Also very important, how do the AI scientific outputs diffuse into the economy, and what are the macroeconomic implications? And here, very interested in thinking about what are the bottlenecks, for example, around clinical trials or like wet lab experimentation that might prevent us from realizing the benefit from AI.
So you talked about kind of the geometry of machine learning problems in science, and also like root node problems in science. And in some ways, if we think about the spectrum of the distribution, it seems like protein folding is the most machine learning shaped science problem that exists. But then everything's across this distribution. And I assume that distribution may be more like a power law than a Gaussian or something like that. But I guess I'm curious, to what extent do you think that stuff is so far from machine learning shaped that it's really only for humans to be doing versus machines? Or is that like, so this is a little bit of speculation on the future, but it's also speculation on what's the current distribution as you see it of machine learning shaped problems in science.
Yeah, I think I'm not sure, like actually, just because I'm not a domain expert like in biology to be able to answer that question. Like I feel like what I would like us to have is like a better evidence base to identify where the opportunities are based on data availability of benchmarks, as well as gaps, maybe areas where we are seeing like a lower than average exploration of AI methods. And then maybe like in those areas, the answer is there's data gaps, we don't have benchmarks, people don't have skills, or it might be that there's some sort of like irreducible complexity or unpredictability to those phenomena, or like a human component or something, or like some sort of like barrier to entry for AI models that prevents the adoption. But I don't know what that distribution would look like, but I would love people to try to like estimate it in some way. Okay, the final one is very straightforward, which is I would like you to add yours. What other evidence gaps are you think are important? Are you working in any of these areas? We are looking for people to interview, like for this essay, like about these gaps. We're looking for reviewers for the essay when it's ready. And actually, people want to get involved in like writing the essay with us. We're also looking for co-authors. So yeah, I just wanted to put it out there. And then, so I've got the last slide and just an observation here. Is it okay if I go over by 2 minutes, Ryan? Yeah. I feel like this is like a big question for me as someone who's like a user of this evidence as well as a generator of the evidence, which is how does the evidence production system about AI and science need to change in order to create evidence about this kind of thing in real time? Because these models have been developed now, they're being deployed now, they're creating impacts now. I feel like the cycle of several months or even years that take us from like thinking about a paper to creating the paper and publishing it It means that the evidence base that policymakers are able to draw on in order to make decisions is always going to be lagging. And my question is how can we accelerate that a bit? And what is the role of automation? What is the role of AI? And obviously, very excited to be talking with you about this today. Like I know you work with SciSciNet, SciGPT, and other tools that might help to contribute to accelerate how these evidence production systems work so that you get this evidence and address these questions in time or using computer science to inform decisions. And yeah, my final idea is something that actually jumped at me reading the AI scientist paper recently, which was that actually, if you think about this, what this AI scientist is, it's like basically an orchestration of AI agents to do scientific research. You have an agent that generates ideas, an agent that reflects, an agent that ranks ideas, an agent that duplicates, an agent that does meta review, an agent that evolves ideas, all of this being orchestrated by this supervisor agent, and this is generating hypotheses. And actually, like this is being used to do scientific research around antimicrobial resistance or drug repurposing, or identifying drug targets. But I was thinking, wouldn't it be interesting to actually use this kind of setup to simulate science? Could you actually like say create different things configured in slightly different ways and see what they come up with? Could you actually have teams where some teams have access to specific data sets or specific tools, and you compare their performance? And some of them you're running these like almost like synthetic experiments, just to evaluate the impact of different interventions, different resources, or different ways of organizing science into the outputs. I think that would be super interesting. And yeah, it's if anyone's interested in that idea, I would love to discuss it with you. And yeah, just the final thing. Science needs AI to tackle increasing scale and complexity. The emerging evidence suspects benefits, suggests benefits, but also lots of evidence gaps. And also, it's not a case of building and you will come and they will come. Actually, a lot of work is required to realize these benefits. And yeah, we have to make the right policies and the right evidence. That's it. Contact details if you want to discuss any of this.
Perfect.